Recombinant factor manufacturing became possible when factors 8 and 9 genes were discovered. By transferring the genes into host cells, scientists created cells that consistently produce high amounts of factor 9 protein. Once the appropriate cells have been identified, the cell line is frozen for storage. For each manufacture run, small quantities of these cells are grown in a series of increasingly larger incubators. Ultimately, they are grown in a large bioreactor with a nutrient-rich but albumin-free media. It usually takes 3 to 4 days before the protein is ready to harvest. The liquid is first filtered to separate the cells and additional impurities. Several chromatography columns are then used to separate factor 9 from other proteins and impurities. The columns are filled with beads that will capture factor 9 while allowing impurities to flow by. The columns chosen have been optimized for binding factor 9. The factor 9 protein is then washed from the beads using specific buffer solutions. The entire process occurs in a controlled environment and is constantly monitored. Finally, a viral filtration step is included to reduce the risk of any viral contamination. The factor 9 protein is now stored at low temperatures. Upon passing extensive testing, it is processed into its final formation and freeze-dried into a powder. The final drug vial is now ready for packaging.